Okay, welcome to week 4 of BIO113. And this week we're going to talk about microscopy, microscopes. We're going to look at a few things. Well, we're going to virtually look at a few things under the microscope and uh, talk about what we're seeing and what's going on, and get down to the cellular level and what's going on with some of these things on the cellular level. So first, let's talk about microscope itself. This is in the actual class that I teach. This is the microscope that we use. And I put this unlabeled picture here for you to reference. I go back and reference, because when we get later on in this presentation, you're going to see a picture of the microscope with arrows and names all over it. And it's going to be hard to see some of these actual parts once they've been pointed out in the images. So I just wanted you to have this to reference. You can go back and look at after we talk about it. But a couple things I'll point out here while we're here, and I'll reiterate these later. Up here at the top, this is the ocular lens. This is what you look through. Obviously, you put your eyes here to look through the sample, which will be down here on this big rectangular piece called the stage, okay? And down at the bottom, here's your light source. It shines up through the stage, through your sample, and goes into these lenses here. These are called the revolving nose piece, where you've got your magnification lenses on here. And this particular one has 4 lenses, and you can actually turn these to get the different magnifications. So I'll talk all about that. You've got on the right here, this big round knob is your coarse focus, and inside, on the inside of that knob, sticking out here, is your fine focus. So you always want to use your coarse focus when you're first bringing your specimen in focus, and then finally adjust that with this fine focus knob. Now, hanging down here from the stage, and I wanted to point this out because it's hard to see later on, this is the stage, the knobs that actually move the stage. There's 2 knobs here. This top knob, I believe, moves it side to side, and this bottom knob moves it back and forth. So it's easier to see on this picture than later on. All right, so let's get into this. Parts of the microscope. And these are compound light microscopes, and I'll explain that a little bit later, but just basic parts, as I talked about. We got the base, which is obvious. This is where the support for the whole microscope. And then you've got your arm, which is, this is actually where you would carry the microscope. You'd put your hand around this and carry it, one hand on the arm and one underneath the base. Body would be this area at the top where it's attached to the ocular lenses, okay? And then your mechanical stage, and it's called mechanical because those stage control knobs that I pointed out earlier can move this thing back and forth, side to side, okay? And the slide holder is this little clip on the top. Let me go back here. You can see a better picture. This little metal clip here opens up, and you put your slide in here, and that will hold your slide while you move the stage around, okay? So, as I just said, you've got this slide holder. So to use it, you'll push this lever, and it kind of opens this little jaw up all the way. And then you would set your slide in there and release the lever, and it'll hold the slide in place. You don't want the glass of the slide to be under any of these metal parts. You want it to be in contact with these in 3 different points. Okay. So here's some more parts. And this is one of the main reasons I put that initial picture up, is you can't hardly see what's going on with all these labeled parts. But so we've got on-off switch, light adjustment dial. So this dial down here turns up or down the light that's coming out through the illuminator. As I said, these are the focus knobs. This is the stage control knobs, and the stage being this piece here. And underneath the stage is 2 things that are important for the microscope. You've got the condenser, which actually takes the light and focuses it through the sample up here that's sitting on the stage. And it basically makes sure that an equal amount of light is coming through all areas of this lens that's underneath here, this stage area coming through the sample. And there's also an iris down there. Basically, you can think of this as like a camera iris that opens and closes to allow more light through or less light through. Regardless, the condenser takes whatever light the iris lets through and condenses it down into what you're looking at on the actual sample. And so I said this is a revolving nose piece. This holds the 4 different objective lenses. Okay, so the shortest one, 4x lens, is pointed out here. And I'll talk about the others. There's 4x, a 10x, a 40x, and a 100x. And you can see they, besides these power names 4, 10, 40, and 100x, they also have names such as scanning, which is the 4x, and they call that because it's the lowest magnification, so you can use that to help find your sample pretty easily. And that's where you always start, is on the 4x magnification. That lens actually has a little red ring around it. Low power, which is your 10x, has a yellow ring, and you can see it's 2.5 times more magnification than the 4x. High power 40x has a blue ring, and then oil immersion, the 100x, has a white ring. You only use the 100x when you're using oil, and only on a sample that's basically under a glass cover slip that's been sealed up. All right. Now, I'll talk about this some more on the next slide. Most microscopes are parfocal, meaning when you get the subject on the slide in focus on one lens, let's say you get in focus on the 4x lens, you can rotate this revolving nose piece to the 10x lens, and it should relatively stay in focus on that one. You might have to do some fine focusing with this fine focus knob, but once you get in focus with one, because these are parfocal, you'll be able to rotate through these different lenses to and still be somewhat in focus. Now, I talked about the objective lenses here on the nose piece. These guys have the 4, the 10, the 40, and the 100. And I mentioned briefly this ocular lens. There's also a lens in here, and it has a magnification power of 10x, okay? So if you're looking through the 4x objective, or if you're on the 4x objective looking through the ocular lens, your total magnification power is actually 4 times 10, 40x, okay? Likewise, if you're looking through the 40x objective lens, you gotta, don't forget about this 10x ocular lens, so your total magnification is gonna be 400 times there, okay? So you can see with these microscopes, the maximum magnification you can achieve is 1000x, all right? So, and I just talked about this, total magnification is the magnification of the objective lens, these lenses that are hanging down here on the nose piece, times the magnification of the ocular lens. Now, this magnification of the ocular lens is always 10x, okay? Anytime you see a question that says, what is the magnification power of the ocular lens? You'll know that it's 10x. Now, these are called compound because they're magnified not only by the objective lens, but also by the ocular lens. So it's 2 forms of magnification. So that makes it compound microscope, okay? And this blue lines kind of show you what happens with the light when it comes through. It comes from the light source up through the condenser, gets condensed down to a fine point on the specimen. Then it goes up into the objective lenses, bounces around a prism, comes back out to where you can see, okay? This reason these 2 lens system is good is because it allows you to see distinguish between 2 points and in a depth perspective to be able to tell which one's on top and which one's on the bottom. But you're limited depending on the quality of your microscope. So if you have a microscope that has a resolving power, so this is resolution is what this is called. Resolution is your ability to distinguish between 2 points. And if you have a resolving power of 0.4 nanometers, it means you can distinguish between 2 points that are more than or equal to 0.4 nanometers. If you have 2 points that are closer than that, you won't be able to tell which one's on top and which one's on the bottom, okay? So, and then I mentioned before, parfocality, specimen stays in focus when the objective lenses here get rotated, the 4, the 10, 40, and 100, okay? So, how do you use a microscope? So, you always start with your microscope on the 4X lens, and you will have the stage all the way down. So you use this, let me back up. Whoops. You'll use this focus knob. You can turn it in a counterclockwise manner and it'll lower this stage down. That's basically when you're focusing it, you're raising the stage up so that it is focused through the lenses. But you always start on the 4X and then you slowly bring this up until your sample comes in focus, okay? So start on the 4X scanning power. You will use your coarse adjustment dial, position your stage about an inch below the objective, and then turn your light on. Make sure your slide is, put your slide in there. And you can actually, when you're looking at the slide, before you look in the microscope, you can see the light coming through from below the microscope. So you can kind of position it a little bit that way on your sample that's on the slide. So you start out that way, and then you can look through the ocular lenses while you slowly raise the stage using that coarse adjustment dial to find it, to get it in focus. Once you get that in focus, you can adjust the fine focus with that fine focus knob, okay? So once you do that and you get it in focus on the 4X, then you can rotate that ocular lens to the 10X power lens, okay? Now remember, we were on 4X and the total magnification there because the ocular lens is 10X, that was 40X. We've now switched to 100X because we got our 10X objective lens and our 10X ocular makes it a total of 100X. So if you're looking through this ocular, there's going to be a little in one of the two oculars. It's only in one of these, so it doesn't mess with your vision too much. There's going to be a little black pointer that basically points to the center of the area you're looking at. So you can actually use this to your advantage to help get whatever you want to look at on a higher power. So you would position, say you're looking at this hydra through the microscope, you could position that little black pointer so it's right above here, right in the center here. So when you rotate your objective lens, this should always be at that center point. You don't lose it when you get closer in, all right? And once you rotate to your 10X or higher, you can use your fine focus. Always after the 4X, you only ever want to use your fine focus, because what happens is you're very, very close to this glass slide with this microscope, with these objective lenses. Even on the 4 or 10X, you're pretty close to these guys. And if you use that coarse adjustment knob, you can very easily just rotate it so that it goes right into the objective lens and cracks your slide and damages the lens. So you just got to be very careful if you're doing that. All right. So let's talk about some aspects of the properties of the microscope. So there, in the question set, there's some questions about when you're looking at an image through the microscope. It'll talk about what you see is like an upside down, image of what's actually on the microscope. So I say upside down, and I'll see if I can pull up this. I'll show you later. It's in the question set. And there's some questions about that. And some of those, for you guys who are strictly online, I'll have to give you the answers. It talks about when you move the slide, a stage to the left, which way does the image move? And it'd be kind of hard for you to figure out if you don't have a microscope to do it with. So I'll have to give you the answers for those. But some things I wanted to point out. All right, as you increase your magnification, you're going to decrease three things here. Okay, number one, you're going to decrease the distance between the objective lens and the actual specimen here on the glass slide. And then basically you can see that pretty easily. These different lenses are different lengths, and this is a cartoon representation of what these look like. But the distance between the lens and the specimen is called your working distance. And as you increase your objective lens to higher powers, this working distance gets shorter and shorter because these lenses are longer and longer, okay? Easy one. So as you increase magnification, you're decreasing this working distance. All right, also, as you increase magnification, you're decreasing your field of view. All right, so when you're on the 4x objective, you can see this entire ring, let's say. But when you switch to the 10x objective, you're actually zooming in so that you're only seeing this much of the original sample, all right? And the same with 40 and the same with 100. So you're actually getting, even though if you look to the ocular, it still fills the ocular, what you're actually seeing is zoomed in so much that you're basically seeing smaller area, okay, just more magnified. So as you increase your magnification, you're decreasing your field of view, okay, and that's what that means. You're decreasing how much of the specimen that you have, that you can see at a time. And you're also, as you increase your magnification, you're decreasing your light intensity. So you're actually decreasing how much light comes through, because imagine if you're looking at this on the 4x lens, and the amount of light that comes through on this whole sample is even across, all the way across. If you zoom into a smaller area on the 10x or the 40x, you're getting less light because you're not able to see the light that was on these lower magnifications. So you're letting less light through. Now you can easily compensate for this by turning your light level up or opening the diaphragm, that little kind of like an eye that opens to let more light in. Okay, so it's easy to compensate. You just got to keep that in mind. As you switch these lenses, you'll have to change that diaphragm and or adjust your light levels. Okay. All right. So we can actually measure this field of view with a microscope on, say, the 4x objective or the 10x objective. You can put a ruler under here and measure the actual distance. And here's what we did. So we took a ruler and we have it on the 10x objective, you can see here. And we put a ruler under there, and you can see, this is metric because we use metric in science. You can see these tick marks. Each one of these tick marks is a millimeter. So from this first one to the second one, it's 1 millimeter. And from the second one over here to the edge, which you can't see, would be 2 millimeters. So the distance across here on the 10x objective lens is 2 millimeters, is your field of view, okay? That's referred to as 2 millimeters or 2,000 micrometers, because there's 1,000 micrometers in a millimeter, all right? So if you say you want to know what you're seeing on the 40x objective, you have to do a little math. You can base it on what you're seeing on the 10x here and figure out what you're seeing. So, you know, on the 10x, you've got the 10x ocular and the 10x objective. So your total magnification is 100x. So for 100x magnification, you can see 2 millimeters or 2,000 micrometers, okay? So for the 40x, we know the total magnification of that is 400x, right? Which is 4 times greater magnification than the 100x. Easy enough, 400 is 4, it's 100 times 4. But what that means is you're getting 4 times closer, okay? So you would take what you see in this 10x objective and divide it by 4, and that's what you would see in the 40x objective. All right, so if we see 2,000 micrometers across here for the 10x objective, for the 40x objective, we're seeing 1/4 of that, or 500 micrometers, right? 2,000 divided by 4 is 500, so 500 micrometers. Easy enough. And you can do it the other way too. If you want to know what you're seeing on the 4x objective, you can take this 2000 micrometers, and 4 is 2.5 times larger than the 10x. You're seeing 2.5 times more than you would on the 10x lens. So you would just multiply that 2000 by 2.5, and you get what, 5,000 micrometers or 5 millimeters on the 4x lens. So now knowing on the 10x objective that the diameter is 2,000 micrometers, right? So if we're looking at a 40x objective, so we know that this diameter now goes from, we said here, it goes from 2,000 micrometers to 1/4 of that, 500 micrometers. So across this whole thing on the 40x objective will be 500 micrometers. And if we know that, and we're looking at this little paramecium here, you can see it takes up about half the diameter, right? So we can say roughly this guy is 250 micrometers. That's easy enough. If I know the distance here based on my measurement, I can figure out just by looking at him how long he is. Now, how wide is he? Might be a little bit trickier, but let's just say you could stack 10 of him high, him, her, it high. So if I know that this diameter is 500 micrometers, then a tenth of that would be 50. So he's probably around 50 micrometers wide and 250 micrometers long, okay? That's a rough estimate, all right? So here's where it gets fun. Now we get to do some math, and I'm sure you guys are really looking forward to doing some math. So what we do here is we'll take a plant leaf. They've got this common name wandering Jew. This plant is, I forget what the scientific name of it is, but it is got purple and green thick leaves, and on the bottom side is all the chloroplast for this guy, okay? And we're gonna, stomata, I'm sorry, not chloroplast, but stomata. Stomata is what opens up to let carbon dioxide in, and that's where the chloroplasts are located, the little green things that do photosynthesis. And then they open up again to let out the oxygen that we are happily going to breathe in ourselves. These are like, if you could zoom in here really closely, they look like a little mouth there. It kind of opens and closes has to open and close to prevent loss of moisture during the heat of the day and stuff like that. But it opens to let in and out gases. And so we're going to calculate the area of the field of view at low power here. So we measured, let's back up, on low power, which remember 10x objective is low power, right? 4x is scanning. So the diameter here was 2,000 micrometers, right? So if we want to calculate the area that we looked at, we know the area of a circle, the formula for that is area equals pi times r squared. What's r? R is your radius. Radius is half the diameter. So if the diameter was 2,000 micrometers or 2 millimeters, remember, your radius is only half that, so it's 1 millimeter. All right. So for area of the 10x objective, we can multiply pi, which we simplify by 3.14, times the radius, which was 1 squared. 1 squared is still 1, easy enough. So the area of this field of view on the 10x objective is 3.14 millimeters squared, okay? So if we looking at this on the 10x objective and we count the number of these stomata we can see, and you're welcome to count this, it's about 50. And for math purposes, I like to say it's 50 because it makes the math a whole lot easier. All right. So we want to know in in this area that's 3.14 millimeters squared, we can see about 50 stomata. So we want to know how many stomata would be in something the area, the size of a quarter. Okay. First, you have to figure out the area of the quarter. So the diameter of the quarter is 25 millimeters. So how do we figure out area? Area is pi r squared. So the radius for a quarter would be 12.5 millimeters, right? So we got pi, which is 3.14 times 12.5 squared. All right. That gets you a number of 4,906 millimeters squared for the area of a quarter. Okay. So we can now set up a ratio and figure out how many stomata would be in that area of a quarter. So we have our original numbers here, number of stomata in our field of view, which we said was about 50. And the area of the field of view, which is about 3.14, not about, is 3.14 millimeters squared, equals x, the number we're looking for, the number of stomata we're looking for in the area the size of the quarter. Okay, so let's do this. Area, our number of stomata in the field of view is 50 divided by the area of the field of view, which was 3.14, equals 15.9. 15.9 equals x over the area of the quarter. So you want to multiply both sides by the area of the quarter, which, what was it I said? The area of the quarter was 4,906. So I'm going to multiply this 15.9 by the area of the quarter to get x, right? So 15.9 times 4,906. I'll get back to this. 4,906 gives me 78,121 stomata in an area the size of a quarter. Okay, easy enough. So I will tell you that the spotlight instruction this week is going to be covering this again for those of you that didn't follow my explanation. And it's a little easier to explain if I've got like a board to draw on, but that's the best I can do here. Okay, so you now have the power to figure out how many stomata are in the field of, are the size of, you know, whatever you want. Okay, last thing I'm going to talk about is we've got this plant called Elodia, and when you expose the cells to salt, a high salt concentration, you can cause the cells to change how they look. And this is because of osmosis, which is the movement of water from a high concentration of water to a lower concentration of water. Okay, so let me explain what I'm talking about. So up here is a normal picture of these cells under a microscope. And you can see the even distribution of the chloroplasts, and here these little green things, and they just look like normal happy cells. When you take those same cells and put them in a 10% salt solution, so 10% salt, we can assume is 90% water. Let's assume the inside of the plant has about 99% water. And if I put them in a solution that has 90% water, it's going to want to move water out of the cell and into the environment to try to make a balance that water concentration or salt concentration, both. I get a balance to that. And so what happens is the water moves out of the cell and ends up causing the cell to like pull away from the cell wall and shrivel up inside, because it lost water, right? All the water that was in here, not all of it, but a lot of it came out to try to balance this equation on both sides. So water moves from an area of high concentration inside the cell, 99%, to an area of a lower concentration outside the cell, which was 90%. All right. And that is all the show for this week. Now, we have, let me stop sharing this. We have, this week we're going to have, same as last time, we got a Labster for you guys to do. We got a question set for you guys to do and a quiz. just like before. And as always, email me with any questions, and I'll get back to you as soon as I can. All right, thanks.